Leading a startup team, whether you're delivering a sugar rush, stocking coffee, or getting a regular delivery of snacks, Office Depot has solutions that fit every startup culture, from getting those first business cards and stationery to ordering fleece pullovers with your new logo. To learn how Office Depot and the California Technology Council have partnered to bring you savings on all of these startup essentials and more, go to californiatechnology.org/member benefits. I'm Daniel Levine, and this is the Bio Report. Perlara is a drug discovery company focused on using model organisms to find treatments for diseases previously believed too rare to cure. Recently, Perlara entered into a drug discovery and development collaboration with Novartis, a deal that included an equity investment and is seen as validating Perlara's platform. We spoke to Ethan Perlstein, CEO of Perlara, about the company's unique approach to drug discovery, why it focuses on the diseases it does, and the significance of its agreement with Novartis. Ethan, thanks for joining us.
Thanks so much, Danny.
We're going to talk about your company, Perlara, its approach to drug discovery for rare diseases, and a recent agreement you entered into with Novartis. First, I thought we could begin with Perlara's approach to drug discovery. You use model organisms that have been engineered to serve as models for rare diseases and screen potential drugs. Can you walk me through that process?
Sure. So we can imagine a drug discovery process where everything that happens in patients has a parallel in the model organisms. And we can start to think about it from the point of view of the registries. So the first step in our process is to model a particular rare disease in different species, in different animal models, yeast, worm, fly, and fish. And the starting point is to understand what are the mutations that actually exist out there in patients and try to model them specifically. And that's where gene editing technology comes into play and allows us to create essentially personalized disease avatars, a fruit fly or a worm that would have a particular patient's specific mutation. We could also obviously make more representative mutations, but we can get this kind of precision thanks to CRISPR. And so the second step would be to understand what are the biomarkers that are displayed in these disease models. And so essentially we carry out natural history studies in these simple animals. And in that way we can verify that these models are therapeutically relevant and then able to be screened at high throughput, where we use the whole animal as our screening substrate instead of using cells or individual proteins. And then the process for us culminates in identifying new compounds that are active in a mouse model. And we know that that's the point at which you could achieve a partnership with a biopharma company if you were to advance an asset, still early stage, but to that point of preclinical efficacy. So that basically sums up the Perlara approach.
What makes this a good approach, particularly for the discovery of drugs to treat rare diseases?
So we think that rare genetic diseases, and especially the rare genetic diseases that are caused by single genes, in other words, mutations in a single gene, it turns out that when you do the analysis across many thousands of these single gene diseases, you're not finding a random set of the human genome. You're finding more disproportionately ancient genes popping up among rare disease genes. And so we think, therefore, that studying these diseases of ancient biology is appropriate in these simple animal models where we believe that the underlying biology is faithfully conserved.
You're focusing specifically on lysosomal storage disorders. Can you explain what this group of diseases is and why you've chosen to focus on them?
Yes. So lysosomal storage diseases are a group of about 50 related diseases that all involve some kind of defect in a part of animal cells called the lysosome. And every animal cell, whether it's a single celled yeast or us, has lysosomes within their cells. People liken it sometimes to the recycling depot of the cell, but I think it plays a much more fundamental role in cellular physiology. And so for that reason, this area of biology is, as I said in my previous response, quite conserved evolutionarily. And it also turns out that from the patient advocacy point of view, because these diseases have been known to medicine, either just as a disease, sometimes for decades, and then with a genetic diagnosis, in the last 20 years, there's a lot of well-developed patient advocacy in the lysosomal storage disease space. So for a combination of reasons, both scientific and from the point of view of advocacy, lysosomal storage diseases, LSDs, are particularly well de-risked, we thought, for our process. And for any kind of rare disease effort in general, because there are a lot of rare diseases for us to choose from as a platform to sort of go to market. And lysosomal storage diseases, for a lot of reasons, fulfilled those de-risking criteria.
You mentioned that once you had a mouse model, you could then partner with a pharma. What's the business model? How far would Perlara expect to take an experimental drug?
Yeah, so right now, the way we see the process is sort of like the Silicon Valley minimum viable product, or MVP approach. So what we found is that it's possible to start from zero to create disease models, and then get to a point of preclinical efficacy, or at least to have a set of compounds that are suited, chemically speaking, for mouse validation in less than a year. And so that's what we've already demonstrated, that is something that not only can work scientifically, but from a business development point of view, in the sense that you can create a minimally viable, partnerable asset, if you will. But the idea for us is that over time we want to hold on to the assets longer, and part with them, closer to IND, where they can get a much higher price relative to the additional costs you have to put in to advance from preclinical efficacy to IND. So that ultimately is the goal, is to want to become a IND marketplace, where we advance assets to that point, partnering with patient groups, and then having many avenues of clinical development. And the LSDs represent one particular avenue, which is working with one of the best pharma companies in the world.
Well, let's talk about that. You recently entered into your first significant collaboration. Last month, you announced a drug discovery and development collaboration with Novartis. This is what I think you'd call a validating deal. What can you say about the terms of the agreement, which did include an equity investment?
Yeah, so I'm not at liberty to disclose all the specific terms, but essentially it's a deal that comprises an investment in Perlara, and also a collaboration agreement where we're going to be working on 3 lysosomal storage diseases together, starting with Niemann-Pick type C, which is Perlara's lead program. And again, the way the collaboration is envisioned is that we would be able to boot up a disease program in the model organism, sort of from scratch, as we did with Niemann-Pick C or NPC, and then advance in a sort of 2-year time frame or less to a point of preclinical efficacy, at which point the asset or the chemical series, in this case, would then be handed off to Novartis for further optimization and preclinical development. So that's how the partnership is and the collaboration is structured.
Novartis is not a company that comes top of mind when thinking about rare diseases. What's their footprint in rare diseases, and what do you think is driving their interest here?
Well, they definitely have a program in SMA that I think is nearing some kind of fruition. So I think that there is an experience on their side in rare diseases. I think this represents a larger opportunity to think about the connections between rare and common diseases. I think we're not the first to see these connections, especially lysosomal storage diseases. But I think that definitely is an animating spirit to the collaboration, is to see lysosomal storage diseases in this case as a way not only to arrive at what Jay Bradner calls definitive medicine, where you would have a real non-incremental, sort of simply palliative effect, and that's what we'd be striving for. And at the same time, you can also target ancient biology in a way that is disease modifying in more common indications that may involve the same underlying biology. Then I think that's an opportunity that that obviously excites a lot of people, not only in rare diseases, but people looking into the rare disease space and wondering what could be their strategy.
The lead program you mentioned is on Niemann-Pick type C disease. What is Niemann-Pick type C? How common is it? How does it manifest itself? And what's the progression of the disease?
And Niemann-Pick C is quite devastating among lysosomal storage disorders. Usually, there's an infantile form, but usually it's diagnosed as a juvenile form, and the kids usually don't survive into adulthood. We're talking in America or the United States at least, several hundred patients, and maybe 1 in 120,000 births is sort of the frequency you see this. And again, these are rough estimates. So it's definitely on the rarer side of rare compared to, say, fibrosis, but not necessarily what would be considered maybe ultra rare, where there may be only fewer than 100 patients diagnosed. And as a lysosomal storage disease, NPC has sort of features of lipid accumulation. In that case, among others, with cholesterol, but it may not be as simple as some of the other LSDs in terms of, well, you just have too much of something that normally doesn't accumulate, and the whole disease flows from there. It appears to be more complicated than that, given that the NPC1 protein is not only an evolutionarily conserved protein, but one that plays a really interesting function in the cell. In fact, it sort of almost completely serendipitously turns out that Ebola virus and that class of virus use the NPC1 protein as the entry point into cells. So all these sort of quirks of biology get illuminated when you start probing any one given rare disease, and NPC is no exception.
And finding a treatment for a Niemann-Pick, does that possibly have ramifications for more common cholesterol and cardiovascular diseases?
Potentially. I mean, people have done GWAS or genome-wide association studies where they've implicated variation in the NPC1 gene with, for example, obesity and potentially other sort of metabolic related and potentially cholesterol related diseases. But again, that I think those are sort of suggested at this point. But they are strongly suggested, and you can look to other examples within LFCs where there are very clear connections between what amount to a rare form of neurodegeneration and a more common form where it's essentially the same underlying biology affected.
And does doing the deal with Novartis give you an opportunity to use larger libraries of potential drugs to throw against these screens?
Absolutely. That's definitely the idea here. So what the kind of, again, one of the animating spirits of the collaboration is that we want to put in vivo models at the top of the screening flowchart. Folks like Novartis have done an amazing job of building capacity to do cell-based screens and target-based screens, but doing whole animal screens, that's our special sauce. So there's definitely emphasis on building those models and then exposing them to different kinds of libraries. We have our own set of commercially available compound libraries, but we can certainly be screening these with their collections too.
And you have, as a preclinical candidate, you have identified a lead. How close are you to entering the clinic with the Niemann-Pick type C disease?
Yeah, so for Niemann-Pick C, for NPC, we have a lead molecule that we refer to as Pearl 101, and we've actually been blogging about it on our blog, and we're actually doing an update pretty soon. We started the series of posts about almost exactly a year ago, and we're still a bit of ways away. Still have a lot of work to do. So there's still some definitive mouse efficacy studies that need to be performed. There's still some lead optimization that has to happen to improve the compound. The compound was sort of born ready in a lot of ways in terms of having oral bioavailability and blood-brain barrier penetrability and other great properties already baked into the starting structure, but it can be improved in terms of potency. So we definitely need to do some more work on the preclinical side, but the goal is eventually to obviously reach the clinic as quickly as we can. But as I say, there's still more development work ahead, and we know that there are obviously always pitfalls associated with drug discovery, no matter what step of the process you're at. But we're moving forward, and we're also characterizing Pearl 101's mechanism of action and hopefully also its target ultimately, which will also help us understand better, how it could be useful more broadly beyond NPC once we've ascertained that it really is its primary indication is for NPC.
You're also working on NGLY1, an ultra rare disease. Does the Novartis agreement cover that as well?
No, so that's separate, and so actually that's part of so that we sort of saw the pharma partnership. If I step back a minute from the business perspective, we saw a pharma partnership as a way to get that validation, as you said, the validating deal. And what was that really validating? Well, it's validating the kind of bottom up joint partnerships that we're calling PearlQuest, that we want to be doing with individual families, foundations, whatever sort of the patient advocacy group, however it comes composed. We want to be partnering with them and do essentially the same kind of process we did with the NPC program, but do it in the context of a bottom-up patient partnership. Again, with something that we're calling the PearlQuest. So we'd say Angle 1 represents an example where we'll be doing a PearlQuest, and hopefully that will be the first of our first batch to come in 2017.
Angle 1's interesting in that it's had some implications in bone growth and cancer and Parkinson's disease. Do you see potential for attracting pharmaceutical interest in that by its other applications?
Absolutely, and I think this is just a trend that we'll probably keep seeing, which is that if you look at these ancient genes that are causing single gene diseases, it's going to turn out that once you fully elucidate the surrounding basic biology, the connections to disease states are going to start to become very apparent, and it won't be sort of an exception that this is going to be a case where you find a definitive therapy for a very rare disease that also ends up being disease modifying in more common indications. I think the exact path for Angle 1 has not been elucidated, but that's part of the thrill of this discovery process, is trying to uncover not only a therapeutic for this disease, but understanding how more broadly useful could that therapeutic be.
We can discuss why traditional farmers find rare diseases attractive, but they still have to get economics to work for them. Are you concerned that some of these diseases you're pursuing would be too small a market for a big pharma? Is that an issue?
It potentially could be, and I think part of one way I think there are two ways to approach that. One is I think we get our unit economics costs really low. If our unit economics are really good and our costs are low, then I think we can sort of say, look, these programs are not going to be hindered by failure legacy costs. And so we can really, I think, potentially be much more transparent about the costs going into developing therapies, and we do aim to make those costs go down. So I think that could be one strategy to sell this idea of investing in rare diseases. And then the kind of follow-up is, once you've initiated that investment, as you start to understand more about the biology of the disease, you're inevitably going to find connections to something more common. I think it's going to be the exception to find a rare disease that if you do something, it has zero impact on any other surrounding, either related rare diseases or any other disease. So I think it's only a matter of time, especially as we develop, a bit deeper insights into human genomes and simply have more human genome sequences around. I think we'll start to see that the connections that start popping up between any given rare disease and more common, essentially more common manifestations of that same broken biology.
Ethan Perlstein, founder and CEO of Perlara. Ethan, thanks so much for your time today.
No, thank you, Danny.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.